Thank you very much for the great talks. I have somebody for Dr. Ingber. Is there any organ that you cannot build?
How much money do you have? I'm not going to build a large bone to look at the effects of gravity and deformation on large bone. Although we've built a small bone. Kit Parker's doing a brain, but it's really neuronal connections. But he's beginning to look at certain wiring connections. I think we're building I like to say we're building living 3D cross-sections of minimal functional units of organs. So it's like a living 3D histological section. But I think you can, we're about to try a lymph node. There should be no reason you couldn't do a pancreatic islet. I think if you think about it right, like what are the key interacting players, and you give them the right microenvironment, they do it on their own. We have not changed the medium that has been used for 50 years with KCO2 or these others. We're just changing the microenvironment. The fluidic environment's unbelievably important in these, often for survival and function. People in the liver chip, and also the endothelium and the epithelium, you have to have both a lot of times to get functionality. We're just beginning to integrate the stromal cells, which are going to be extremely important for certain functions. And immune cells. We're just integrating the immune cells into these chips. But I think you could go pretty far with most organs. Is there an organ that you're thinking of that you want to challenge me on? No. Okay. Kit Parker's done heart, skeletal muscle, airway smooth muscle, vascular smooth muscle. But he's not. It's a much simpler system where it's just sort of muscle on a flexible membrane and they contract. But now he's integrated an endothelium underneath so that we could connect them to our systems.
And do you integrate also the stem cells or the satellite cells and see how they just...
He hasn't yet. See, it's the level of question, right? You're going to ask certain questions. The thing I like to say is that, and I said it in the mini review, or the commentary in Cell, is that you can learn things assumptions that you have that are wrong by this because it's synthetic. So I don't know if I said this, but in the pulmonary edema paper, one reviewer said this should not be published. It's overly simplified. There's no immune cells. And the other reviewer said, this is really interesting because it's a synthetic system. They just discovered you don't need immune cells for pulmonary edema induced by interleukin-2. And that's true, and it got published. It took a lot of fighting, but it got published. So I think you're going to ask questions in a model, and if satellite cells are important, you're not going to see the response. So then you got to add satellite. But if you do see the response, maybe satellite cells aren't as important as we all think.
I'm just kind of curious if you understand the mechanism of why flow has such a positive impact on what you've seen, like the flow of whether it's blood or whatever underneath.
Well, flow, that's been studied for years in endothelium, but also basically flow and stretch both can affect cells through specific molecular pathways, and there are many types of transducers. Flow in the kidney, there's that primary cilium up top, acts like a little cantilever arm with the flow, and that activates calcium signaling, and it's very important for function. Stretching on the cells pulls on integrins, activates every type of signaling pathway you could kind of think of, all the way to nuclear to transcription. So flow is like, I often said years ago that if you found extracellular matrix before growth factors, you would have called extracellular matrix a growth factor and growth factors fine tuners. And if I had just been able to pull on cells on matrix first, I would have had pull was a growth factor and the others were fine. There's like a three-body problem. They all regulate each other. But the mechanical forces are regulating signaling. They're also changing cytoskeletal structure, nuclear structure, and all interplays to regulate attractor switching for the people in the systems biology crowd, because you're hitting multiple nodes in the network at once. And so you're able, and evolution is selected for structures being linked to signaling, and the signaling is, much of it's on the cytoskeleton.
So is there a difference between like endogenous versus exogenous mechanical forces? So in the alveoli, it's because of the diaphragm, the pressure difference, while in peristalsis, it's because the cells are themselves contracting, similar with heart. Does that come into it at all in terms of how the chip works?
As I said, God, 30, 40 years ago, that the main stress the cells feel is the cell-generated force, because if that were weaker, they'd be popping off the matrix every time that something stretched. So it's like a winched-in bow and a bowstring, and the cell is pulling, and now it's taut, and now it feels its environment. Now, bone cells in vivo sense very small deformation, microstrain, but pretty high frequency, just moving around. Your aorta, it's 1 hertz once every heartbeat, but it's pretty big strain, 5, 10%. You take those cells, isolate them of everything, put them on a dish, they have the same sensitivity. So nobody understands that. It has to do something with how they adhere, the cytoskeleton, the matrix, so we give the right physical environment. The cells are doing their pulling and whatever they were doing. It all comes together, and somehow the rhythm and their pulling, it somehow either, you know, it's just harmonious, and they do their own thing. It's a wild thing to watch.
So I think what I really enjoyed about this session is that each of you sort of presented a new frontier in our understanding of living systems. So whether it's using information in a totally new way that we already have, or moving from nucleic acids to proteins, or moving into a three-dimensional space where we can incorporate mechanical information. I guess that's what the organizers had in mind. So I guess my question to the three of you is, what is the next frontier in the type of information or the way that we incorporate data into our understanding of living systems and biology and disease and how to treat it more effectively?
You start.
All right, I guess I'll start. So I think model systems are obviously growing in terms of sophistication and relevance, but I think another direction is how can we more carefully use humans themselves as a model system? I think you know, we are capturing data from humans better and better. We have Apple Watches and wearables. That's an extremely crude type of way of doing it. Obviously, I think we'll be able to get better measurements in the context of what people's behaviors are. Maybe measurements, you know, with little droplets of blood without using Theranos. But maybe there'll be others. So I think more measurements on humans is another direction. I think you know, humans might be the ultimate model for humans. So that's one way to think about it. Obviously, the NIH is betting this will happen and is funding precision medicine kind of programs this way, trying to get 1 million volunteers to actually give up a lot of data on themselves, including blood tests, urine tests, microbiome, all of that. So hopefully in all those measurements, there is something there. But the kind of implicit kind of thing, though, is data by itself does nothing. It doesn't move anywhere, right? It takes someone to ask the right question of that data to come up with an interesting discovery and maybe even something useful for patients. So data by itself does nothing. You need to put some energy into it to release the knowledge. What do you think is next?
I would agree with that. I think the big challenge is making really good phenotypic measurements across many scales, from molecular to, you know, things like using Apple Watches and so on. And because the underlying collection of the genetic information now is, again, the first approximation to solve problems. So if you can get enough high quality, sort of detailed, high resolution phenotypic data across many scales across populations, you can probably make all kinds of really interesting associations that some would not be expected.
I told Atul after listening to the talk, I'm applying for a job at his place. So, but you know, I think
if you bring the money, sure.
Yeah, I think the you know, the idea of where future research needs to go. Now, talking I give that the human is the best model, and the amount of info you can get out of what people have already analyzed, which is, again, looking under lamplights a little bit, but still so rich that you won't be able to, you know, use it all in our lifetimes. But there's the things that people have never done studies on is really where a lot of interesting things lie. And I hope these organs on chips could let human experimentation go at that level. But at a more fundamental level, I think being able to look, a lot of people here I know do single cell analysis. In my view, that's going too far, but you want to do it, but you want to do it in the context of the population in a physiologically relevant microenvironment. So I like these chips because you could potentially be looking at 1,000 cells by analyzing readouts of each one while a process is happening over time in a lung while it's breathing versus not. Whereas a single cell on a chip, and we've done this in the past, with microcontact printing, can tell you what happens stochastic, the on or off switch, all or none between states of individual cells and temporal, but it doesn't talk about how the collective behavior is really regulated in a physiologically relevant context, which involves physical, it involves gradients, cell-cell contacts, and then there's the hormonal sides of these things. And with these sorts of systems, you can control those. And so I think having systems where you could study both the granular and the integrated collective behavior in a physiologically physical context, that's required to go to the next level. I don't see NIH supporting that at all, actually.
But there is this tension, right, between more and more reductionism and more holism, right? Whether it's at the patient level where we don't believe in populations anymore, we want the individual, right. but the individual cell might be too much. I mean, there is no right answer still here, right?
We have to go both ways.
Yeah.
It's like building the tunnels in Dark Ages, Middle Ages, whatever.
Dr. Butte,
Sure.
I'd like to ask you even about bigger data. So how do you coordinate, say, with other institutions, so IBS and Providence, or I'm from Colorado, the University of Colorado system is trying to do this, or take it to Luxembourg where the whole country's storing data. How do you look at all that to optimize outcomes?
Yeah, so first of all, I still have to solve my state's problems first, okay? But I hear what you're saying. So I think what we're trying to do is just show with examples what we're going to do with this data. I mean, I think you can build repositories, but I'll be the first to admit they're useless unless someone does something with the data. So our attention right now, we spent the first year, we put at least one time, we got a dump of all 5 medical record systems into one. So we're building the kind of apps. I showed you 10 different bullets there for just different ideas that we had because we have 10 campuses of University of California. So we're kind of opening this up to all the campuses and try to get these use cases. There are some interesting use cases out there. And when we get good use cases, then I think we're going to attract other parties of interest and really kind of integrate the data. Now, the whole country, if you follow the electronic medical record field, they say harmonization is hard. And I'll be honest with you, I don't think it's actually that hard. Of course, I'm building on 5 years worth of work before I got there to UC, but really, when I look, there's basically 3,500 things we do or measure on patients, okay? 3,500. Everything else is a long tail, okay? But 3,500 things we do or measure. The typical drugs, the different heart rate, blood tests, stuff like that. If you kind of line them up, it doesn't take very long to get them lined up. And so let's solve them partner by partner instead of countrywide kind of thing. Because when we talk countrywide harmonization, everyone throws up their hands and says it's not possible, it's not possible. But if you start with smaller groups, smaller collaborations, I think it is possible. I think that's kind of the approach. Maybe we'll add a few of these kind of groups that want to use or use case some of these kind of apps that we're building or applications of this data and go from there. Let me just give you throw out some numbers here for a second. So we have 14.5 million patients. So I have 20,000 with breast cancer, okay? 20,000 with breast cancer, fully EMR data. I mean, it's crazy if you think about it. There's a particular drug used in breast cancer. I'm not going to mention the name of the company or the drug. A particular drug that's monoclonal antibody used in breast cancer that we have 2,000 patients that have been treated with this drug. Now, if you look at the pivotal trial for that drug, it had only 300 patients. So now I have 8 times more data on that drug in the University of California than the pivotal trial from that biotech company. So that's kind of interesting, right? Because I know what their trial showed, obviously got approval based on that, but now I have what people are calling real world evidence on this drug, right? What actually happens with this drug? Is it better or worse than the trial? So this is interesting to pharmaceutical companies, biotech companies. thrills them and also scares them because I can build models now of where I think the drug is working or not, right? Pay for performance and things like that. So I think there are going to be all sorts of downstream models. Science is one of them, but just improving operations, reducing errors, and also just really cost effectiveness research to figure out what is actually working in medicine or not. So it's a long answer, but I think we're just starting to figure out what to do with all this data.
So to continue and moving to your vision for 2019, that we essentially have real data, continuous monitoring of individual patients, the microarrays is a standardized way to collect information. And so what do you think we need to standardize where it comes to electronic health records or any other type of information?
Yeah, so people really have problems with the quality of the data in electronic medical record systems. Everyone, especially physicians, say, they're entering this data, they think it's the lowest quality there is. But actually, I would bet it's the most expensive data in the country right now, because we pay doctors to type in stuff, right?
Yeah, exactly.
Per byte, it's probably the most expensive data in the United States, is electronic medical record data. And I know it's got problems. I know there are these short, really wide people because we put heights where weights are and weights where heights are, so you get these weird BMI calculations. We know all these errors. You can find a bunch of these. You can fix them. We know these common errors. And remember, what my philosophy is not to pick on the data. I know it's going to keep getting better, and it's going to get more plentiful, right? So what I really say is that, let me put it this way. I know it's full of problems, but think about it this way, right? As a medical doctor, I'm barely a medical doctor at this point, but if I have a patient in front of me, right, on a gurney, let's pretend right here, actually legally and legitimately, I can print out that electronic medical record in paper form, put it with that patient, send this patient somewhere, and that other place can read this and save that patient's life today, right? So it's good enough for that right now, right? To save lives. I mean, it's good enough to take care of patients. So I know there's problems with this data, but we know where a lot of the problems are. It's always going to keep getting better. So why wait for perfection, right? So the point is, it's great data as is. It's expensive data. The average byte of data is probably never looked at again, right? Unless a patient comes back, which on average is rare. That data, you spend a lot of money collecting that data, none of it is used. And that's kind of weird if you think about how many billions we're spending on collecting this data, how little effort is going immediately downstream of that. So that's what we're thinking about.
So
yeah.
It's a great point to say, why wait for perfection? But are you scared that you'll then convince everybody that there's no need to improve?
Yeah, that's a great question. Can we do that? So I think, that's kind of also the beauty of our University of California system. And so the way I kind of spin this to people is we have these 5 medical centers, and we're kind of all prima donnas. We all think we're doing something the best. So just take any area of medicine. I don't know, take pneumonia care or something. And just look with the data. Maybe 3 are doing something better than the other 2, right? So can we learn from 3 and teach 2 and just keep rotating, right? Pick something else. Okay, well, 3 are better than 2, or maybe 1 is better than the other 4. And that actually is, I think, what they call a learning healthcare system, right? We're going to learn from this data. No experts involved here. I mean, teams have to be ready to adapt and to perfect their care. I mean, we've got to get through that kind of culture change too. But that's the ideas. The data keeps getting better, but we know medical care actually has problems. Can we use it immediately to try to fix something there? That's.
Hi, Atul Butte. I have a question for you. So you've been talking about aggregating microarray data, and you've also been talking a lot about personalized healthcare. So many diseases, including preeclampsia, which you mentioned, are thought to be caused by different biological changes manifesting in the same phenotype.
Yeah.
So I was wondering, could you use publicly available data to identify these clinical subtypes, or at the same time, do you worry that the clinical subtypes are going to confound your analyses, like whereas it's like preeclampsia, yes, no?
Yeah. So the idea of using public data, or at least microarrays and now public data to find subtypes, it goes back almost 15 years. So Ashley Zadeh had one of these great papers, a seminal paper in Nature in year 2000 on B-cell lymphoma, where it's like a 50-50 split based on microarrays, and they could show a survival difference. So that's a crude clinical difference driven by molecular. I think a lot of the breast cancer classifications now are from microarrays, or at least from gene expression. So I think we're there. I think what you can go further is to use public data. I mean, to not even have to run the microarrays yourself. I think we are in that kind of role. I'm definitely not the only one doing that kind of thing, though. So one example is pathway diagnostics. So not pathwork diagnostics, not pathway genetics. Pathwork diagnostics, they're defunct now, but they created the first FDA-approved chip, microarray, to look for cancers of unknown etiology, like metastases. And if you look, I mean, they couldn't sell enough. That's a different story. But you put a cancer on there, and they could tell which of 15 cancers metastasized, because sometimes you find a metastasis first, you don't know what the primary was. But if you look at their work, they actually developed a whole FDA chip based on public data. They downloaded data from Geo, they got the chip design, and then they tested it, I think, with Pittsburgh or someone with a lot of cancer samples. So I think you can do that, and I think you can come up with diagnostics, and I think you can get them even FDA approved to take them further. So I think it can be done, but I'm not the only one doing it for sure. There are many, many doing that now.
So can you have data from the same patients over time?
That's a great
so that you can look at dynamics of progression and maybe get towards causal gene?
Yeah, this is great. I mean, so yeah, question about how much you get like longitudinally.
Right.
Obviously, we love our EMR data because it's longitudinal, but it's rare to get molecular testing. Now, I think there are certain cancers where, based on protocols, you can get, for example, you might get a biopsy, they might get some pre-surgical type of chemo, and then you might get another biopsy, but that maybe is 2. You may never get 3. Kidney transplant or organ transplant, if they're on a protocol, sometimes they get protocol biopsies, you can run those, people have been doing that. But you're absolutely right, it's rare to get that. Now, take that to an extreme, you have like Mike Snyder getting like narcissomes, you know, the Snyderome thing. He's doing like another 50 or whatever. There's a bunch of people doing this, obviously, all around us here. I mean, so I think that's a kind of future story, to get more of those. But it is still an expensive experiment to see how much you get out of just measuring everything repeatedly on people. I admit, I'm going after low-hanging fruit with the public data. It hasn't been plucked yet, right? Maybe it's faster.
I have a question that we discuss a lot at ISP, and that is, how do we now integrate, for the entire panel, how do we integrate everything? Because we have done with really, it's the epitome of mechanistic and understanding physiology, and then with big data, Atul, you said, you know, there's no mechanism, still useful. So what's the next challenge if somebody wants to integrate everything?
You want to try starting?
Repeat the question, please.
How do you
integrate?
How do you integrate a mechanistic study, which is very complex on few patients, versus big data, which doesn't have mechanism? Yeah, and I think one day we'll have to do that.
Yeah, I mean, listening to his talk I was trying to think how you can counterpoint, you know, if you can get those specific patients and then use these type of chips, analogous to my finding a genetic subgroup to do a clinical trials with that subgroup. You know, when he has his maps of the fireworks progressing,
they are fireworks,
right?
exactly.
Can you get those? You know, if you have every child gives iPS cells and he gets a gene microarray every year through his life, you get your dynamic data. We can make chips things. It may be possible, but I think you'd have to do design a study with a specific group of people where you can see whether, I mean, you're doing it on a population average, and now you want to do personalized.
That's right.
And so at one level, you could cheat by trying to find the average group who fits in the middle of the bell curve, right, and see if that goes. Then you could expand that by looking for a random group and see what that spectrum looks like.
Right.
See if you can go back with your data and pick them out. I think it's doable.
Yeah.
Anyway, that comes to mind off the bat.
You have thoughts? I mean, the way I would think about it, go ahead. Yeah.
Well, you know, I think this is a long way off, but I think at some point, and I suppose this is the ambition of systems biology, fundamentally, right, is to build a model, some sort of predictive model. And how far do you have to get with data before you can build some, usefully predictive model where you can throw anything at it and fit it in?
I think the thought is a sort of deterministic model that would be predictive, but you might just be interplaying, complementing a bit of your population-based data that's correlative but predictive with bits that you understand mechanistically that are more, if you like, deterministic or even nonlinear based. But I think it's going to be a mixture of all these sorts of things.
Yeah, I mean, it is a truism. How much do we really understand even today in medicine, right? I mean, we have a lot of correlations, not much, right? It depends where you think of where we are on the iceberg. How much, do we know 10%? Do we know 90%? I mean, we're probably close to like 2-3%, I'm guessing. But I mean, in terms of integrating all this into a database or into this, I mean, to me, when I look at my lab and what we do, I think the hardest part of what we do now is not the code. Of course, we're on the cloud. Of course, we have clusters. All that's doable. It's not hard. The hardest thing to do now is figure out what's the question to ask. Okay, I mean, that's what really makes you a scientist and not just
And
an
that's
engineer.
been true in the history of science
Yeah.
since the first
So-
rock was pushed.
Absolutely. A couple of junior folks came up to me and asking for advice right after the talk. The one piece of advice I give to undergrads or high school students is always learn how to ask good questions. Everything else could be outsourced today, okay? But not asking good questions. That keeps you in business as a scientist. Don't even get me started about outsourcing. I have another whole hour talk on that. How we outsource our mice and stuff. So, but if you ask good questions, so here I think that's what we're struggling with. I mean, okay, now we have 14 million patients. What's the question do you wanted to ask here? The killer question. I'm not saying I have all the answers, but in my lab, we never write a line of code. We never start a line of code unless we know what's the question we're asking. Otherwise, you're going to write code and you integrated everything, and then still nobody uses it. The build it, they will come kind of era is over, I think, for informatics. So now we as computational folks have to ask the questions. We can't just enable everyone to ask the questions with our tools. There's no one out there. So we got to enable ourselves first, right? Put on our oxygen mask first, ask the questions before assisting others. All right.
Okay, so then one more last question.
So just following up on that comment.
Sure.
I think it gets back to the lamppost analogy.
Oh, yeah.
You're asking the questions that you have intuition to ask.
Yeah.
And we're missing all the rest. And I just want to ask from someone in the computational sciences, is there a day coming when we don't need to know what to ask?
Yeah.
Where we can just throw the power of unbelievable computation at it and let it ask everything?
So I don't know. I'll try to answer this first. I don't think it's going to come. I mean, someday, obviously, there's supervised and unsupervised machine learning. Let's put it this way, right? And unsupervised, you get interesting clusters, but they're more interesting if you had a question in mind before you ran the clustering, right? I mean, so it's always you're doing all this work for some reason. But if you're starting with no reason, you do find a lot of correlations that people implicitly already knew, okay? My favorite example is I have computational folks looking at all this lab data. And they said, you know what, there's this thing called a CBC, complete blood count with differential. That means they looked at the white cells. And it's almost exactly correlated with this thing called CBC without diff. We've known that for 100 years, right? But you're going to get 1,000 years of these, if you didn't actually ask the question. So I am still a fan of supervised stuff because you're in a hurry in life, right? You got to get to something, and if the tools can help you, you can get there. So I think the last day of science will be the day when the system can come up with the question by itself. Then that's the day you turn off the lights and leave. But that is truly the last. I don't think it's there yet. It's not going to be there for a while. I don't know.
Do you think? I mean, if I just push it a bit harder, there's at some level you can reduce this in a practical way to pattern recognition, right? And we only have so much capability to do this. And so one by one hypothesis testing is great and it's got us a long way, but we're looking at 14 million patients and 100,000 data points per patient. This is not even within the realm of possibility to process.
But he didn't say
I'm
hypothesis.
not saying you do it.
You said questions.
It's a question. Let's go
is there
further.
a problem we want to solve, right? If you have
what's the unmet need you're going after, right? Exactly. That's never going to be outsourced. I mean, the day that's outsourced, you're done. Okay, science is done. But you always start with some need here, right? Some unmet need. You see a patient suffering, or you see some biology that needs explaining, some unexplained finding. You can wait for the software to get to that one, but you're going to die before it does, right? I mean,
Can't asking the right question be the need?
Yeah, that is the need.
In at least a limited context of these very large data sets, recognizing some patterns that give you a starting point for asking questions.
I think that is still the hardest thing in science, is learning what is the cool question. What's the question everyone wanted to know the answer to? No one realized we could ask it today, whether it's the tools or the data. I think that's what really makes you a great scientist. I think is to learn how to ask those questions. And you see those questions when no one else is there. It's intuitive. I'm not saying there's a systematized way to get there. I don't know. Maybe people have a systematized way to get to good questions. But I think that is what makes you a good scientist, I think.
All right. Any more questions? Good questions? You can leave it to machine learning.
Yeah,
okay,
only good ones.
Okay, then I'd like to thank the panel again and all the speakers for today.